Let's wait for one more minute as more people are joining in, and then we'll start. Hmm. Okay, I think it's time. Let's begin. Hello, everyone. Welcome to our first All Protein Dialogues webinar of 2026, Upgrading Alternative Proteins with Fermentation-Derived Ingredients. My name is Ravi, SciTech Specialist in Fermentation at GFI India, and I will be your host today. It gives me great pleasure to welcome Anupriya, Eve, and Shubhankar to our webinar. These innovators are paving the way for driving the next step in the evolution of alternative proteins. And we are grateful that they could take out time from their busy schedules to share their expertise with us today. But before we begin, allow me to introduce the Good Food Institute. We are a non-profit think tank working across the globe to build the alternative protein sector through three key streams of intervention: science, policy, and industry. How do we do that? Well, through a network of affiliates spanning across the globe, from Brazil to Europe to Israel, India, and Asia Pacific, and with the recent addition of Japan and Korea. We engage with a wide array of actors in the food systems and drive the change from within. All Protein Dialogues is a collaborative initiative co-organized by GFI Asia Pacific and GFI India with the goal of fostering regional partnership that drive meaningful, impactful global innovations in the alternative protein sector. And today's panel represent a good mix of Asia Pacific and India. Now, before we jump into the webinar, I have few quick comments for our audience. Today's seminar will run for total 90 minutes. The speakers will present a maximum 15-minute presentations, followed by a 25-minute long panel discussion at the end. On the bottom of your screens, you will see chat and Q&A buttons. We encourage you to use the chat function to introduce yourself to other participants. However, please make sure to direct your questions in the Q&A window. Also, try to mention who your question is directed to in the specific Q&A section. And you will also have an option to upvote questions you would like to be answered, so we can prioritize those that are most relevant to our live audience right now. So without further ado, allow me to invite our first speaker on the virtual stage, Anupriya. Anupriya Sundarajoo is an industrial biotechnologist specializing in application of enzymes to enhance the flavor and functionality of plant-based beverages. Her expertise lies in the practical use of enzymes for improving texture and flavor, protein modification, and developing proof of concept for new plant-based drinks. She's particularly focused on leveraging enzyme technologies to create high-quality, consumer-acceptable plant-based beverages. And not just that, her previous work included enzyme applications in the home care industry, as well as optimizing microbial cultures for enzyme production, for bioremediation and biodiesel applications. Anupriya, thank you so much for joining us. The virtual stage is yours.
Thank you so much, Ravi. Thank you so much for the introduction. And I would also like to express my gratitude for actually inviting me to be part of this webinar. It's really a pleasure. So let me just quickly share my screen before I start. Okay, hope you guys can see my slides.
Yes, it's visible.
Okay, let me just switch to presentable. All right, here we go. So hello, everyone. As introduced, I'm Anupriya. I am from Novonesis, part of plant and food team, currently focusing on enzyme application for plant-based beverages. Right. So before we jump into the presentation, topic for today, I would like to take this opportunity to introduce Novonesis, who we are and what we do. So Novonesis is actually a result of a merger between Christian Hansen, which has over 100 years of experience with cultures, and Novozymes, which is a global enzyme leader, a combination that now brings over 100 years of expertise in biosolutions. Novonesis produces biosolutions that has broad application in various fields, for example, bioenergy, household care, animal health, including food industry as well. To name a few segments within the food industry, we have cheese, brewing, baking. Today, the focus would be enzyme application in plant-based beverages. Now, when I say enzymes, what are enzymes, right? Enzymes are actually biological catalysts in the form of proteins. They are found in nature in every living organisms, bacteria, fungi, even us humans. Enzymes are fully biodegradable, but they are not living organisms. Sorry, enzymes are actually proteins that are produced by living organisms to improve the rate of reaction that happens within living organisms. So to jump into the topic for today, we are going to be looking at how enzymes can be utilized to improve texture and taste in plant-based beverages, particularly oat and rice beverages. When it comes to processing plant-based beverages, there are a few challenges, especially when it comes to creating high-quality drinks. Some plant-based raw materials are hard to process and break down due to high starch and fiber content in these raw materials. Certain raw materials also lack natural sweeteners, which may result in a bland plant-based beverage if added sugar or artificial sweeteners or certain flavorings are not added in. And when it comes to processing plant-based beverages, sometimes the texture can either be too thick, too thin, and watery or grainy. Plant-based beverages are also known to have a bitter or unpleasant taste if they're not processed properly due to the natural phenolic compounds or phytochemicals that are present in plant-based materials. And when it comes to the raw material quality, if the consistency is not there in terms of quality, then you will also face consistency issues when it comes to processing your plant-based raw material to produce high-quality drinks. Are you seeing my slide move?
No, Anupriya, it's still the same slide.
Okay.
Processing
No
plant-based.
worries. Let me use the cursor in that case. All right.
Yeah, works.
Okay. So here we have a look at the process flow for production of plant-based beverages, specifically oat and rice, right? So typically what we will do is we'll have the raw material in either flour format or the grain format itself. And then it could be either mixed or grinded in water at high temperature. As I mentioned earlier, some of the plant-based raw materials do have high amount of starch and fiber. Now, what happens when you introduce starch and fiber with water and high temperature? Well, the starch can gelatinize, the fiber can actually end up holding a lot of water, and this results in buildup in viscosity of the total slurry. The next two subsequent steps are where enzymes come into role to improve the texture as well as the taste of the plant-based beverages. For texture improvement, we can use enzymes such as alpha amylases to break down large complex starch molecules into simpler molecules in order to reduce the viscosity and also improve the texture of the overall plant-based drink. Now, once the starch molecule is broken down into simpler components, those simpler components can then be converted into simple sugars through enzymes such as maltogenic amylase or glucoamylases, which allows to produce plant-based beverages with varying levels of sweetness without the addition of added sugar or artificial sweetness. Upon completion of these two steps, you are pretty much ready with the plant-based milk that has been extracted from your raw material. The remaining steps are pretty much just polishing your slurry to produce a finished good that is ready to be packaged and sold off in shelf. So decanting process pretty much just removes all the insoluble fibers that are present in your slurry. You go through formulation to improve the stability of your product, UHT treatment for microbiological control, followed by homogenization, and then finally packaging. Let's take a deeper dive into how enzymes can improve texture in plant-based beverages. For a start, let's look at oat milk. When it comes to processing oat milk, these are some of the raw materials that can be utilized. You have natural oat kernels. You also can use dehusked heat-treated oat kernels or dehusked heat-treated oat flour. Now, although these all are essentially oat, but when it comes to the approach of processing each of these raw material, it may slightly vary simply due to the nature and characteristic differences of each of these raw material. Oat is a raw material that has starch, and it accounts to about 50 to 65% of the flour, and the bulk majority of the starch is made up of amylopectin. If you recall the slide earlier that I mentioned, high amount of starch and fibers can actually pose a challenge when it comes to processing plant-based raw materials for beverage production. Alpha amylases actually help break down the starch into simpler dextrins and oligosaccharides, which results in a drop in viscosity. It's also important to highlight that dextrins can actually act as fat replacers, which improves the texture of your overall plant-based milk as well. So enough talk, let's maybe have a look at how alpha amylase actually helps with this drop in viscosity. So I have a video here. There are no audio in this video, so when I start playing, you will see the visuals moving, so just observe. So if you can look at this video, you can see a very thick slurry that is trying to be mixed in a big stirrer tank. Now the enzyme is being added. You can see that within seconds of adding in the enzymes, the thick, viscous slurry is now much more easier to stir, which will allow much more homogeneous mixing and much efficient production, right? So that's the power of alpha amylase when it comes to reducing viscosity. Okay. Now, let's move on to a different plant-based raw material, rice. Southeast Asia is popular with rice. I mean, it's our staple, right? We eat rice on almost daily basis. There are diverse rice varieties that are available in Southeast Asia that can offer unique benefits. Rice in general is rich in carbohydrate and they are also abundant in starch. Certain varieties of rice are also known to be rich source of dietary fiber, rich source of antioxidants and polyphenols, for example, brown rice and riceberry. Similar to oat, rice also has a lot of starch and it can also be gelatinized when it is going through the plant-based beverage production process. Alpha amylases can also be utilized in this context to reduce the viscosity of rice slurry to produce rice-based plant-based beverages. Now, with the reduction of viscosity, you can also improve the yield of rice drink extraction using alpha amylases that can reach up to 70% depending on the raw material that is used. Here are some of the rice grains that we have tested in the past with alpha amylases to extract higher amount of liquid out. So the higher the amount of liquid you see, the better the extraction process. If alpha amylases are not used when it comes to extracting rice drink, then you will end up with a very thick, gloopy slurry, which will be very challenging to filter and further process to produce rice-based drinks. So these are the benefits of using alpha amylases when it comes to producing oat and rice-based beverages. They help you reduce the viscosity and also improve the yield of the plant-based beverages. Now that we have taken a look at how alpha amylases can help improve texture in plant-based beverages, let's have a look at how enzymes can also adjust sweetness in plant-based beverages. Remember this slide that I had shared earlier? And I had mentioned that we are able to utilize two enzymes, such as maltogenic amylase or glucoamylase, to produce plant-based beverages with varying level of sweetness without the need of added sugar or artificial sweetness. How, you may ask? Well, the secret lies in the mode of action of those enzymes. So when we look at maltogenic amylase, it's actually an endoamylase which cuts dextrins into disaccharides such as maltose. Whereas glucoamylase is actually an exogenous amylase which cuts dextrins from the terminal ends to produce glucose. Maltose actually leads to a mild sweetness increase, whereas glucose leads to a higher sweetness increase in plant-based beverages. As we know, maltose has a relative sweetness level that is slightly lower than glucose, whereas glucose's relative sweetness level is far higher. Hence, when you generate a lot of maltose in your plant-based beverage, it will have a very mild, pleasant sweetness profile. Whereas if you're looking to produce a plant-based beverage that has a nice, sharp sweetness, then you would go for a glucoamylase to produce high amount of glucose in your plant-based beverage. Let's look at this graph, which shows the production of maltose using two different amylases as shared earlier. So here we have glucoamylase and maltogenic amylase. When glucoamylases are used, the amount of maltose that are generated is small compared to what is generated with maltogenic amylase. The reason being due to the mechanism of glucoamylase, which cuts from the terminal end of dextrins, producing monomers of glucose. Why do you see some amount of maltose still present? This could be due to halt of your hydrolysis before glucose could hydrolyze all of the dextrins into glucose. So whatever that is left out could be some amount of maltose, and this could be the amount that is produced. Whereas when it comes to maltogenic amylase, its key function is to produce disaccharides from your dextrins. Therefore, it can produce higher amount of maltose in the plant-based beverages. So that pretty much brings us to the end of this presentation. Just to wrap things up, we have looked at how alpha amylases can reduce viscosity in oat milk and rice-based beverages, as well as improve texture and yield. And we also had a look at how two different amylases, particularly maltogenic amylase and glucoamylase, can help with producing plant-based beverage with varying level of sweetness without the need for added sugar or artificial sweetness. In summary, enzymes actually enable process and quality benefit in plant-based drinks production. Use of enzyme actually helps to simplify your production. You are able to adjust the sweetness to match targeted consumer preferences, as well as adjust texture and beverage viscosity to match different needs. There's also a possibility to create a range of different plant-based beverages from simple raw materials. So in Novonesis, we have the Verdera series of enzymes for specifically plant-based beverage applications. So if you're interested to know more, feel free to reach out to us. So that's all from me for today. Thank you very much.
Thank you so much, Anupriya. Yeah, enzymes are indeed these tiny little boxes of magic, especially love the way that you showed in that tank, like how immediate the effect was. Once again, thank you so much for taking us through your work on plant-based milk. Super interesting.
Thank you, Ravi.
Thank you, Anupriya. Okay, so now it's time to invite the second speaker. Now I would like to invite Miss Eve Samyuktha. She is the founder of 7030 Food Science and Technology, a food tech company focused on building practical, scalable solutions in alternative proteins and fermentation. Founded in 2020, 7030 develops functional plant and mycelium-based foods with an emphasis on nutrition, affordability, and real-world adoption. The company works across research and product development to bring better protein to everybody's everyday's diets while reducing the environmental footprint of food through science-led innovation. Thank you for joining us, Eve. Yeah, just give me a second. I'll pull my screen up and yes, take it away. Take it away.
All right, great. I can't see the slides yet, so I'll give it a few seconds. All right. Hello, everyone. Thank you for joining me today. I'm Eve, the founder and CEO of 7030 Food Science and Technology. We are a fungi-focused biotechnology company working in the space of mycelium-based systems. At a very high level, we sit at the intersection of two things: biomass protein, meaning fungal or mycelium protein as a product, and fermentation performance, meaning how well fermentation systems actually run in real industrial conditions. So our work is not just about creating new biological products, it's also about making fermentation itself work better, faster, and more reliable as we scale up. Can we go to the next slide, please? So in our company, we work where fungal biomass meets the performance characteristics of fermentation. Our core expertise is in fermentation systems that are specifically designed for mass manufacturing. Many biotech companies, work on the R&D phase quite well in small shaking flasks, but we can see that the process really breaks down in larger tanks, sometimes even at 1,000 liters capacity. So we work on focusing on 3 practical metrics inside existing fermenters. So number 1 would be yield. And by yield, I mean how much of biomass do we actually get out from the fermentation tank? The second metric is speed, meaning how fast can we produce this biomass? And number 3 is consistency, meaning how similar each batch is to the previous one produced. Now, this might seem a bit boring, but the last one is also an area where in scale-up, you usually fail. These are the 3 parameters that directly determine cost, scalability, and whether the process is actually commercially viable. Next slide, please. Yeah, apologies, there's a tech delay in each slide. So we started in 2020, and we started as a food company. Our original business was building plant-based ready meals direct to customer. This used our generation 1 soy-based protein alternative. We were not doing fermentation back then. We made real food products. We had B2C customers. And while we were operating in this environment, we needed to scale large really quickly. And this is beyond usual research settings in a startup environment. When we started doing that, we encountered the classic problem of scale up that most startups face, which is cost. We couldn't provide the unit cost parity to real meat. So that's when we had to go back to our drawing board and understand why soy-based protein is not giving us the cost even in scale. And from our research and actually talking to GFI in those days, we encountered that there is actually a better way to produce high quality protein. And that pushed us to pivot from plant-based protein to fermentation, biomass-based production. And since then, we have been focusing on creating our own novel solutions for biomass as well as biomass performance technologies. Next slide, please. Our broader vision is that industrial fermentation is becoming a core manufacturing platform. And this is not just for food. We can see on the news that it's also for materials, chemicals. As Anupriya mentioned, for enzymes, this is the core production mechanism, and also for several other industries. But for this platform to truly scale, performance has to improve without making the systems more complex for the users. So our vision is to build the biological infrastructure that enables this. So this includes optimized biomass production aids, which we call as growth enhancers, specifically for fungal strain biomass, and also the practical process know-how. So this is to make the yield come out in a much better consistent fashion, and also make the whole cycle inside the reactor faster. Next slide, please. Our first core product is the proprietary fungal biomass protein. So this is a mycelium strain that we have optimized specifically for industrial fermentation systems. So over here, we take low value carbon sources and convert them into high value protein, edible protein. This mycelium has meat-like structure, and it is useful both nutritionally and functionally. And nutritionally, as in it gives all the amino acid profile that the body needs, and also functionally provides the texture. So it can be used as a primary protein source in food, as well as a structural ingredient to improve texture and the mouthfeel. So it's produced using standard food grade fermentation, and our fungi strain is also GRAS approved. So it functions quite well in existing equipments and workflow. That means that companies do not need to redesign their factories to use it. This would be probably a best replacement for TVP, texturized vegetable protein, or TSP, texturized soy protein. Next slide, please, Ravi.
Yeah.
The fundamental problem we keep seeing across fermentation systems is that, thanks. The fundamental problem that we keep encountering is that after inoculation, when we add the starter culture, there is often a long lag phase where the organism is alive, but not really growing. It's stagnant. This delays the actual production. Even later, biomass growth often slows down before the fermenter reaches its actual capacity, the real capacity, which means that there's a lot of potential for higher productivity that's actually wasted. So to compensate this, a lot of companies, what they do is just increase the glucose feed or just run longer batches. And we know that glucose is the most expensive part of any media or culture for biomass production. So this shoots the cost up, and there's also a variation between batch to batch. So at industrial scale, these efficiencies directly reduce the profit margins, and it also makes the production unreliable and hard to plan. Next slide, please, Ravi. So our new product line, which is not technically new, this is something we bumped into while we were trying to make our own biomass grow faster. So while scaling our fungi production, we realized that early stage growth was the biggest bottleneck. You know, it's not a unique bottleneck. Every fermenter company has this. So we developed a second product line, which is a growth enhancer. So this growth enhancer is a powder, as you can see in the picture. It's a biological additive designed specifically to support filamentous fungi production from the moment of inoculation. So this actually minimizes that lag phase that we saw in the previous slide and helps the system enter exponential growth much, much faster. So far, what we are seeing, it's about 3 to 4-fold, but we are doing our scale-up trial. So we're excited to see what tweaks we would need to do, but we stand by 3 to 4-fold so far. But the application we have tested it on is specifically for filamentous fungi. It would be interesting to see what happens in yeast. We haven't done those studies yet. When we do, you can I want to say that find me on LinkedIn, but I wouldn't say that at this point. I will be happy to share my email later on. It works by supplying key signaling molecules and essential nutrients that improve the growth efficiency. So that's the primary primary effect of the growth enhancer. We're still on the last slide, Ravi.
Oh, sorry. Yeah. Moving.
It's okay. It's okay.
Yeah.
The growth enhancer slide, Ravi.
Mm-hmm.
Oh, great. Technology never disappoints. The result is the shorter fermentation cycles, as I mentioned before. And the wonderful thing is that companies that make biomass or companies that want to improve are specifically involved in improving the productivity at the in the downstream. It's excellent for reducing the need for glucose feeding, because glucose is where all the cost comes from when we have a sudden order and we want to increase our batch time, reduce our batch time, produce more. This is where the growth enhancer would be a fantastic add. So it can be added directly to the existing media. So far from what we have found out, there's no specific redesigning the process that's needed. It's literally an add-on into the existing fermentation process. Now we go to the next slide, Ravi.
Yep, moved. Take a couple of seconds.
Yeah, great. So the core team is myself, Dr. Arulmani, who's the research director. He leads all the scientific development. Sai Geeta is manages our operation, but she's also closely involved in commercialization. And Uma is our lab manager. We do travel quite a bit from our location in Bangalore to other places, specifically for finding the good CDMO partner for mass producing both our biomass and growth enhancer. So we would say we are pan-Asia in our operations and manufacturing. The market right now, we're targeting India to begin with, and later on moving maybe into exports. And that concludes my presentation for today. Happy to take questions later on any aspect, or if someone wants data of our scale-up studies, happy to share that. If you guys want a copy of my presentation, happy to share that too, or just want to chit-chat, happy to do that as well. Thank you.
Thank you so much, Eve. Thank you for that wonderful talk. Especially loved how you evolved your whole product category, starting from plant-based to fermentation and now fermentation enhancement. And I think that's also something I would like to highlight to all the audience, that as we are growing in this alternative protein revolution, we realize that it's not siloed. It's not only plant-based, only fermentation, or only cultivated meat. There is a spectrum of technology. These three are core technologies, and there's a spectrum through which we can find these hybrid products. And maybe the answer lies somewhere in the middle of all these technologies. And that's basically why we were trying, that's what we're trying to highlight through this session. And thank you, Eve, for very well putting it together.
Yeah, excellent. I do want to thank GFI especially in the year 2021-22, when we were pivoting. It was the fermentation report that really inspired us to move from soy-based to biomass production. So I'm happy to circle back and do a webinar. So
thank you, Eve. All right. So with that, now I would like to invite our final speaker, Shubhankar. Shubhankar is the CTO of MyoWorks, where he leads technology development and product development for the next generation input materials for cultivated meat industry. With a background in mechanical engineering and hands-on experience in biomedical engineering and instrumentation, Shubhankar brings a multidisciplinary approach to designing scalable, sustainable solutions for cellular agriculture. At MyoWorks, he drives product innovation from concept to deployment, helping accelerate the future of ethical and efficient protein production. And he's also a grantee of JFI's research grant program. So welcome, Shubhankar. The stage is all yours.
Thanks, Ravi. Really appreciate GFI for putting this together, for actually trying to share ideas so that this kind of cross-pollination can happen. So let me just quickly share my screen. So Ravi already gave you this fake glowing introduction. So let me just dive right into what we do at MyoWorks. So MyoWorks is a cultivated meat inputs technology. What you're seeing in your foreground right now, in the background, I guess, is a scanning electron microscopy image of our mycelium. So after it has grown and everything, it is actually, when you zoom it in, these little hypha of 1 micron across create these fibrillar spongy structures where cells can sit and grow in three-dimensional environments. And that's what we're going to be talking about here. So roughly, I'll first walk you through the process where we first take our fungus, we ferment it so that we get biomass. So similar to what you have seen with Eve's company doing, where they make biomass, but their journey then pivots from us, right? We take the biomass, we wash it, we treat it, as you would expect with mycelia and with fungi, that there are a lot of anti-nutritive and other like just bad tasting uh, things around the hyphae itself. So you want to wash it, you want to treat it so that it's balanced and it's edible. You mix it with edible binders, and we have made some strides in making the beads in proprietary technology so that you can make the beads in really small sizes up all the way down to what the microcarrier expectation is. When you look at when you look at the biopharma industry, where they use companies like Corning or Cytodex, there are companies that already have these set protocols for what a microcarrier should look like, have these set systems, what cell culture on adherent beads look like. So we want to make sure that that's complied with these microcarriers. So we take that, we make sure that they are properly rounded off, clean, sieved, then we wash them, we dry them, we sterilize them, and then they're ready for packing, shipment, and sales. So that's what we're envisioning to do right now. But let me, before that, walk you through our product itself. So right now, what we sell is the MyoScaf 2D cell culture kit, which is a small 12-well plate with mycelium discs that are fermented in situ, so that we can first understand what the cell mycelium interaction looks like. So we provide these to companies. They seed their chicken muscle cells, mouse muscle cells. We've had beef, we've had fish, we've had shrimp. People will seed that, and then they will check how the proliferation occurs on our material. And once that occurs, they can do all kinds of testing. So this is what it looks like under the hood, or inside the cell culture hood. And you add the chicken muscle cells, or in this specific case, we added mouse muscle cells, and they were able to proliferate and create three-dimensional structures. So what does mycelium provide here, and why we chose mycelium is quite specific. If you look at the sort of large Venn diagram, or the universal set of materials that are suitable for cell culture, you have like the plastics, which are quite common, and people from this industry or with cell culture background know that you're using tissue culture plates with some kind of or ECM attachment. So you're using collagen, fibronectin, or other animal origin or recombinant materials that have animal origin, because these substrates are naturally what the cells are attaching well to and proliferating on. Then we looked at plant-based and other fungal materials, and that's where this idea that funguses are actually slightly closer to animals than they are to plants, right? Like this fundamental biological truth rings true for us, because then it has certain attachment motifs available for the cells to actually create attachment, which is what we saw in our lab work as well. And there was, I think there was one more paper out of an institute in Singapore that did something similar, and they found specific attachment motifs in certain fungi, and they were able to catalog it and categorize it. So instead of just isolating the attachment motif, here you get a sort of two-in-one offer. Otherwise, in other plant-based scaffolding material, what you have to do is get a plant-based structure, which is usually a carbohydrate, or a polysaccharide, where you have either alginate or you have other cellulosic materials, and then you graft on animal-like motifs onto it. So whether it's RGD or other peptides. And that grafting and chemistry usually ends up using cross-linkers that are quite toxic. With fungi, you get this sort of protein top coats and the structural fibers all in one. So now you just have to figure out how to grow it all in one properly. So that's the sort of intellectual background. Now let's talk more about what we have done. You can see here a micrograph of mouse muscle cells really happily spreading and proliferated on our material. We've also done cell adhesion trials on these 2D scaffolds, where you seed a set value of cells and then you flush it after 4 hours with a whole bunch of media, and then collect the supernatant, check what is actually flushed out, what is actually adhered, and then like the sum balance is the adhered. So you get a good value. So you get up to 60 to 70% of attached cells on the scaffolding material. And interesting to note here is this is after the washing steps and the cleaning steps, because if you have, the layer above it contains a whole lot of sort of anti-nutritive or, sort of damaging cytotoxic materials that would otherwise kill your cell. But when you do it this way, then the cells are actually able to attach and proliferate. So we're able to see a good proliferation curve across 7 days. Some representative images where you see like these blotches growing and colonizing the material. Um, in addition to this, at MyoWorks, what we're also focusing on making three-dimensional structures. So these three-dimensional structures can get made after the fermentation. We're able to shape it into shapes that have the right kind of porosity and structure. And then you're able to seed the same cell. So the last field image that you're seeing is a shrimp-shaped scaffold where we seeded chicken. I think this is with mouse muscle cells, but they were able to proliferate and they were able to create growth immediately. So what we're looking to do is use what mycelium gives you naturally, which is the taste, the texture, everything that Eve talked about, and then add the taste, the texture specifically, and then we add the cells for taste and nutrition and animal sort of familiarity in palate, and wherein you can create a sort of cost-effective material, that people can eat, that has a good nutrition profile and actually can be successful. We've also, of course, tested our material with a whole array of food relevant cells. So these are primary chicken muscle cells that we have developed or kind of stored in house. We actually sell these materials as well. So there is a lot about cross compatibility here. Is what we've learned across our years with cultivated meat. We incorporated MyoWorks in 2020, which was supported through a government grant, and then we also were supported through a series of GFI grant and some other things along the way. So we see that there is good proliferation, and we're able to show that this is a substrate that the cells like. And these are some representative images from the lab because it's easier to explain. What we eventually want is, chunks that are fully proliferated. So another form factor, just before I go to the next one, is these small microbits. So these are 1 mm by 1 mm across, where you see cells attaching and proliferating. And what we're looking to do eventually is create these micro beads. So you have the mycelium-based balls that are about 150 to 200, 300 microns. They're soft, smushy, and fibrillar. And then the cells attach to it and proliferate as we do it in a stir tank bioreactor. So I don't know. I think I'm slightly before time, but I hope I've covered things. And I'll also have the recency effect because I think Q&A is right after this. So like or after the panel. So we will talk more about it. Thank you, guys.
Thank you so much, Shubhankar. Yeah, it was super interesting to see how you're pushing the boundaries in cultivated meat using fermentation-derived ingredients, mycelium applications. Super interesting to see. Again, thank you so much for sharing your insights, your experience, your research with all of us here today. And I guess that's another way of presenting, or it's a great example to see how fermentation-derived ingredients, here is mycelium, can help in upgrading or bringing in the next generation of cultivated meat, adding more value towards the cultivated meat research discourse, which is going forward. So thank you again, Shubhankar. All right. So with that, I guess it's time we move to the next part of the webinar, which is the panel discussion. But before we begin, I would like to remind the audience to please take the next 1 minute to post your questions in the Q&A box as we set up the video feed with everyone on your screens. Also try to write who your question is addressed to. It could be individual or it could be the whole panel as well. It's up to you. Okay. So we'll take a minute to set up the whole video here. Meanwhile, we'll start the questions. Okay. All right. I think that we have some questions in already, and all of you are here. So let's begin. My favorite part of which is the panel discussion. So let's begin with a question. I mean, it directs to all of you. In your respective applications, what was the single hardest challenge that you were able to fix with fermentation-derived ingredients from a product perspective? So, was it the texture? Was it the taste? What was or maybe like an off note or something that you've seen? So maybe we can begin with Anupriya.
Yep, sure. So the question was, what was
The most difficult challenge
right?
that you were able to fix with the fermentation-derived ingredient. So here, like using enzymes, you mentioned about two different categories of texture as well as flavor. But what, in your opinion, was the most difficult for you to fix using enzymes?
Mm, I guess I would say each of it has its own difficulties, right? When it comes to texture and taste. At the end of the day, when you create a plant-based beverage, you can't just say my texture is great, but my taste is really bad, but you deal with it, right? So it comes hand in hand together. But for me, I believe texture would be one of the most crucial steps when it comes to producing plant-based beverages, simply because that's one of the early stages. And if you mess that up, the rest of the stages are just gonna have a huge domino effect. So for me, I would say with the use of enzymes and the right combination of enzymes, I would say, because in this case study, I've only shown how alpha amylases alone can improve texture. But with the use of other plant-based materials, there is a possibility to consider using combination of enzymes to improve the texture of plant-based beverages, which then would help improve your overall production process. So yeah, that would be for me.
Okay. Eve, what are your thoughts on it?
Yeah, I would say that I remember up until 2022 and even a little bit in 2023, I felt when we were working with TSP, the texturized soy protein, to make the meat alternatives, there wasn't much hope about what we can do about cost, because most of our suppliers were based in mainland China, and that's, the land of getting the cheapest soy protein, right? So the costs always seemed very daunting. And yeah, I mean, I've learned my lesson is that the customer says they care about quality and they care about taste, right? But when it comes to the, end of the day, even if it's in the premium category, they all fight for the cost. It's cost, 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 cost. And, at one point I was like, really, I'm pretty sure you have the money. You just don't want to give me. I took it personally. But then later, it was a reality because margin in food products are already so slim. And, the end customer base is still evolving. So they don't see that as a, how to say, it wasn't exciting for them to have this if the cost did not meet what the number they had in their mind. So I asked them, okay, what if I give you a product that's very similar to the meat products you're using, but it's equal to your meat products price? They said they're like, I need to taste it. I don't want to change. I said, what if it's 30 rupees per kg, just for example, or 50 rupees per kg? They said, can you send the sample tomorrow? So I saw a very different shift in how they were, thinking. And I think this is only possible with mycelium, because with mycelium, you don't need the land to grow the agriculture products to put it into the extruder and make the texturized protein, right? So it's kind of like one step further where you can use something that's already there in nature. Fungi is already producing that with absolutely no light. and we use low value carbon sources and convert them into high value protein. So that has a lot of hope for us in the future that we can compete significantly in the cost. So that's why we find it very hopeful versus being in TSP area.
Yeah, that's a great insight. Shubhankar, although cultivated meat is still, I would say, like very nascent as such from a product category, but is there any kind of specific product challenge that mycelium-based scaffolds can help?
Yeah, so I think that I'll just echo what Eve said, that having two things at the same position by nature is what I saw as a good opportunity. Again, this is maybe from a research lens than from a first sprint, business sense, but the economics eventually will follow the research and the first principles. Is that if you're making a material that has the surface chemistry that the cells like, and at the same time offers you some degree of texture and structure, then you're solving two problems with one thing. And that's something that I saw within mycelium, being food safe, being available, being able to tweak it and tune it to your whims, to find strains that work for you. There are so many more options that open up, as opposed to the other options of making edible scaffolds would be, polysaccharides, grafting, chemistry, plant proteins, or any of those challenges. I think that their mycelium has a unique advantage.
Actually, my next question goes back to you, Shubhankar. So you mentioned about fine-tuning, fine-tuning required for such kind of technologies. So what was the hardest part of, what was the hardest to tune? Which property was hardest to tune in case of application of mycelium as a CM scaffold?
So I think we're still, it's early days, and we have a lab level where, say, we're at TRL 3 or 4. Things change when you scale, especially so is totally correct. So when you're able to do it at a small lab scale, and then you're like, okay, I'll just do this thing at 10,000 liters, it just doesn't work the same way anymore. The way the mycelium expresses proteins changes, the way you are able to process it in sterile setups changes, the equipment changes. So we are very much in a work in progress, where we're negotiating this idea of we have a TRL 3, 4 ready product. We already have been putting this out there. Now, how to take it to scale? And that's been the sort of main challenge that I think, when it comes to fermentation-derived products in general, is if you're doing anything other than just a very specific kind of expression, where there's already a pipeline for purification, a very known way to go from shake flask to stir tank to stir larger and larger stir tanks. It just takes a lot of sideways thinking and trial and error, and that's very much where we're at right now.
Amazing, thank you. Okay, my next question is for Eve. From the audience, which someone asks, how does the cost of your growth enhancer compares to adding more glucose in the process?
Yeah, I answered that with my email.
Oh, you did? Oh, okay, amazing. Sorry.
Yeah, but I can say that it is I would say it's not comparable, as in it's significantly lower. So I'll leave it at that. It is more nuanced than just saying it's super low, buy everything from me. So I'm happy to share the data package with you. I believe I can see the question actually in the back end. Yes, to Nitish Bhagat. Yeah, if you can shoot me an email, I'll send you some data.
Right. I think it's in a similar fashion. There is a question for Anupriya about the application of enzymes. So would the enzymes you mentioned for the texture improvements in plant-based milks say can also be used for plant-based meats, as far as I understand, say for the slurry you create before extrusion?
For plant-based meat, I
Yeah.
mean, enzymes at the end of the day are specific molecules, right? If you have the substrate, it's going to be working. So if your plant-based meat utilizes a high starch content material, then yes, most probably you should be able to use the alpha amylase to bring down the slurry viscosity a bit.
But I mean, so alpha amylase, sorry, just not my domain, was curious. So you mentioned three products, right? Like, and it
Yeah.
was largely, so the first product is just a viscosity reducer.
The alpha amylase, yes, it can be used to reduce viscosity pretty much and improve textures a bit.
Okay, sweet, sweet. And then the other two are the sweetening ones.
Yes, that's right.
I
There
got
are also amylases, but they're
Mhm.
just different in terms of the mode of action.
Okay, no, I got a little worried who wants to make sweet meat, but that's all.
Okay.
There's a market for everything.
Yeah.
That's true. And actually, there are a couple of questions regarding this, Anupriya. People asking for various applications. There's another one, if I may ask. Does Novonesis has enzyme solutions that help break down anti-nutritional compounds during the production of plant-based products?
Yes. So they're not covered in this presentation at this round, but we just recently actually launched an enzyme that can particularly break down some fibers that can cause bloating issues in soy protein particularly. So yeah, we do have that enzyme.
Amazing. Yeah. So I guess it's whoever asked this question, I guess it's time to go to Novonesis's website and see if there's
Yep.
the product that Anupriya diverted you to.
Yeah.
Amazing. Okay. So my next question, again, going back to a little bit on the nutrition side, uh, does the fermentation-derived ingredient that we're adding also adds or affects the nutritional profile of the final product? Maybe again, we can begin with you, Anupriya, and then we can go to Eve and maybe Shubhankar at the end.
Yeah, sure. So I would say for plant-based beverages, yes, addition of these fermentation-derived ingredients such as enzymes can actually alter the nutritional profile of your finished good. So for example, in this case where we had a look at how different enzymes can be utilized to generate different sweetness profile in plant-based beverages, ultimately what you are doing is you're utilizing the starch that is naturally present in the raw material, right, to produce glucose. Nevertheless, although you did not add in any sugars, there's still going to be some glucose. There might be some minor changes to the glycemic index of your plant-based beverages. However, I guess the whole benefit here would be the fact that you are not adding in any sugars or artificial sweeteners, but rather maximizing your raw material to produce products with possibly cleaner labels, right? So, yeah.
Makes sense.
Mhm.
Eve?
Actually, I would need you to repeat the question. I got lost in glycemic index.
My question was that how does the fermentation-derived ingredient affects the nutrition profile of the final product?
Right. Yeah, someone asked a similar question. It's like, what do you do in downstream processing once you have the mycelium out of the tank? So our downstream processing is way simpler than you would have for the plant-based or soy-based meats, because along with TSP, you actually had to add a lot of flour, a lot of it was insane process that I remember. For mycelium, since the texture is already there, it's pretty much neutral tasting. It doesn't have any flavor, it doesn't have any smell. So it's a very simple process of adding fats and some natural flavors. So in terms of nutritional profile, this is the only one that I would say, on the amino acid profile ratio, it's better than some of the animal protein, specifically compared to chicken. So yeah, so nutritionally, I would say it's superior. But yeah, time will tell what we end up adding. Maybe not completely clean label, but we'll see.
I see. Okay. Shubhankar?
Yeah. Okay, sorry, you were saying something.
No, please, go ahead.
I'll So for cultivated meat, I think if you have a scaffold that adds a good amount of protein, just increases the nutritional value. Because if you see some of the other disclosures that have come from the US FDA products, so I think Upside and a few others, these are public domain documents. So their protein content for their cultivated meat products are like 18%, 19%, because the cell itself is quite naked, like there's just water and a little bit, like what the majority of the high protein content that you get from your meat is because of the extracellular matrix protein that are secreted, like collagen and stuff, which is on the surfaces. So if you have a good scaffold that encourages collagen secretion, and if you have the scaffolding material itself, which has a good nutritive value. So again, I'll compare this with like cellulose or with other polysaccharide based scaffolds. If you which is not like a pure mycelium product, you don't have the ability to add protein in. You can then use a textured soy protein product. That's also another avenue to add protein to your final product. So then that 18, 19% number can actually go up to 30% plus.
That's amazing. That's great. That's actually a really good addition to the overall protein intake or the
Correct. I mean, so
quantity.
especially with like Indian, granting and other like food security type of angles, I think everyone is worried about, well, is this stuff actually adding protein content, period, like a percentage protein? They're not even at that. So it will help fortify that the percentage of protein. Yeah.
Well, Shubhankar, I have another interesting question. I mean, it's from your technology part. So the question is, the first image of mycelium scaffold that you showed has uniform pore size and the cellulose size. I believe that it was grinded and binded together afterwards. How do you create the uniform pores? Because I have tried freeze dry, salt leaching, calcium carbonate, and other tools for this.
I think I'll take this on email because there's like hundreds of different pore creation technologies available there. I think they all rely on some kind of like phenomena of lost material. So whether you use salt, you use fibers, you use something that can be chelated later on, um, and mixing is always a virtue. So I think better to take that offline. Yeah,
Totally.
because it really depends on the strain, on the application, all kinds of things.
Yeah, great. So the person who asked this question, please send a LinkedIn to Shubhankar and he'll be happy to answer their questions. Okay, next one's for Eve. So the question is, greetings from Singapore. Thank you very much for all the panelists. Eve, you highlighted that one bottleneck in filamentous fungal bioprocess is the pronounced early lag phase after inoculation. From an industrial perspective, what online or at-line approaches are currently used to monitor early growth dynamics and physiological transitions during this lag phase in filamentous fungi, given their morphological complexity?
Yeah, I answered the
This one as
question.
well? Oh,
Yeah.
okay.
I did.
Okay, I'm using a different
That's
font.
okay. That's okay. Yeah, but I will say a little bit is that this is something usually the bioreactor machine, the tank sellers or the makers of the equipment itself, they usually have some proprietary software that either they make in-house or they import just the software for the online monitoring. But we are working with a company that makes a specialized software. So we are facing some challenges in the monitoring part. So yeah, happy to share more info on email. I keep saying happy to share more info on email. There will be an NDA that you guys need to sign. So yeah, but I'm trying to be as open source as possible. So yeah.
Really great. Thank you. Okay, now I'd like to shift gear and ask if we tap into the consumer part. So, what kind of consumer perception challenges have been really surprising for you when you have used, say, a fermentation-derived ingredient in the final product? And I think, Anupriya, maybe we could begin with you here. As enzymes are still a biotechnology tool, a fermentation-derived tool, and consumers as such may not have enough visibility into what goes into the process. So how So what have you seen from consumer perception when they know about enzymes being used in a process or in a product?
At least within the Southeast Asia region, I don't see enzymes being labeled in plant-based beverage at this point. And the enzymes are typically not carried over into the final product because they will go through an inactivation step to inactivate the enzyme. You do not want to have residual enzyme activity in your final product because then that will affect the overall stability of the product. Therefore, I don't really see a lot of resistance nor acceptance, to be honest, in terms of use of enzymes for plant-based beverages, possibly because of the lack of awareness when it comes to how these plant-based beverages are produced.
I see.
So yeah.
I see. Ignorance is bliss sometimes, but
Yeah.
but still, I'd say enzyme technology, anyways, is quite well established and has been existing for really long. Unlike mycelium and cultivated meat, biomass fermentation and such. So maybe that's also a reason for this. Yeah.
Yeah.
But I would also like to ask, Shubhankar, would you like to go next?
No, actually, I had a question for Anupriya, if that's okay. Specific to what she mentioned on the viscosity reduction, does it like the fibers that they act on, do they all get like reduce in their size, molecular weight? Like, what is the sort of physical manifestation that leads to this viscosity reduction?
So when we speak of alpha amylases in this context, right, what happens is it basically works on starch molecules. So your starch molecules are made up of a lot of glucose units that are linked by
Close.
bonds, right? So this alpha amylase pretty much targets those bonds that are linking those glucose and they will cut them. So what happens is you have a large molecule and then these enzymes work on this large molecule and chop them off to produce smaller molecules, right? So these smaller molecules, I guess they have lesser water holding capacity, similar to fibers as well. When you break the fiber down to smaller molecules, the water holding capacity is lesser. Therefore, the viscosity drops as well.
Okay, thanks.
No worries.
Great. Okay, so if I may circle back on the last question, Anupriya, so do you think when it comes to, and I guess it also extends to Eve and also Shubhankar, do you think when it comes to consumer, what would be the advisable approach, or which approach do you think would be better? Do we communicate only the functional advantage that this fermentation-derived ingredient provides to them, say functional nutrition? Or we just say that, hey, this product or process is being involved in it. So what kind of narrative do you think is better from a consumer perception?
Being a consumer myself, I guess for me at the end of the day would be how much does this particular ingredient that is added into the product benefits me ultimately, right? So if there is a narrative that tells me this particular product was produced with a fermentation-derived ingredient, which benefits you in X, Y, Z, then I'll be like, oh, great. I have a fermentation-derived ingredient that was utilized, fantastic, but I get to reap these benefits for myself. And I guess when it comes to consumer point of view, that would ultimately be the main drivers that would push them to consider to purchase these products, right? So that would be the narrative I feel like possibly consumers would appreciate more. Yeah.
I see. Eve, what do you think? So enzymes are being processed anyway, like Anupriya mentioned, many of them are processed. Sometimes these are not mentioned in the final product anyway, but mycelium being incorporated into the product here, what do you think? What kind of approach would be good to go?
Right. I mean, consumer perception is it's such a even a bigger bottleneck than regulatory, I would say, because even the plant-based ones are facing this challenge, right? When we were doing direct to consumer in the beginning, before the biomass, right? We didn't actually highlight that we make plant-based meats. We put plant-based meats in the meals. We wrote it in very fine letters. And we didn't communicate too much to the consumer. What we focused on was this meal plan or this ready meal will help you get 80 grams of protein, or this much fiber, has no saturated fat or certain saturated fat. So it was more macro focused. It didn't talk much about that you're eating fake meat, basically. To be honest, we did this because I personally didn't buy. I wasn't a good consumer, even though I don't eat meat or dairy products. Okay, I hate saying this. I wouldn't buy mock meats. I'll buy tofu and stuff. So, but the customers we had were very protein focused and macro focused. They didn't care where the macros came from, as long as the macros quantities are met. So I guess the narrative should be not one size fits all for a consumer base. If you say Indian market, it's not a monolith, right? Each community has its own characteristics. So you need to know what bubble you're selling to and focus on that messaging to that crowd. So if it's the fitness crowd, focus on the macros, right? If it's a weight loss crowd, focus on fiber and calories, because people who want to lose weight, they do calorie in and calorie out, and there needs to be a calorie deficit. So you show how this is going to make them feel full and they can achieve the calorie deficit. So I would say it's better to focus on the community of users versus focusing on, you save this much water and it's better for the environment, it's great for the animals. It's none of that is about what is it, how am I benefiting it as a human. So that's a lesson that we have learned, but we might be wrong. Maybe saving animals is what everyone wants to hear. So.
Thanks, Eve. And Shubhankar, considering that cultivated meat in itself has enough questions floating around, when we incorporate mycelium, and again, another fermentation-derived ingredient which has not been, I say it's still relatively new into the overall narrative. What do you think? What could be the communication route? How would you talk to a consumer that, hey, yeah, this is better for you, basically?
I don't know. So since we make the input ingredient that goes to someone, this is expressly because we don't want to answer that question. This is our business strategy. But if I was to answer, everyone with 5 minutes and a mouth is an expert. So let me just give my 2 cents. It's the thinking is that words are expensive, time is expensive. So you say in the fewest possible words possible. Like this is used in politics, used in marketing, used in anything, right? If you can say something in one sentence or less, and you can just say it over and over again on radio ads and print and telephone, that's how you're going to sell. I think the best way is to just do what Eve said, like tasty, fast, clean, food, protein, like one word. So this is with mycelium and fermentation, and it saves cows, and that's our problem. And we're nerds. We enjoy this, and yeah, it's just we are a minority. We're not going to sell like this.
Okay, thank you, Shubhankar. Yeah, that was honest, let's say. Okay, so I think we have completed our 25 minutes, but I have one last question before we part. So this is, I'll first start with Anupriya. So there's a question regarding uh, how SMEs, or how smaller companies, so Novonesis is being a giant, a biotechnology giant, how do you see that these smaller companies which are at the bottom ground working and developing the plant-based product can collaborate with larger companies? Like what kind of approach they can take in bringing more innovative products to market?
That's a very interesting question. I guess when it comes to SMEs working with large giants, right? There are different channels that they can possibly reach out, because not all large companies would be able to directly service you. Uh, so there could be distributor channels that you might be able to reach out in order to gain access to these large giants. Or the other way around would be to be able to come up with really innovative products that definitely has promising potential that these large giants simply can't overlook. So
See.
that's another way.
Amazing.
Yeah.
Thank you. Because Novozymes, or rather Novonesis, sorry, my bad. So
No worries.
Novonesis has basically dominates the biotechnology, biosolutions market, and there are many companies who look for biosolutions. So yeah, so it does bring that. I'd say that the dynamic is not is relatively new when it comes to smaller startups popping up with new products, completely new products, and looking for direction from a large company, which typically look for large volume customers, like B2B, such as Novonesis. So it does create, I would say, like a power dynamic and basically a distance between these two. Whereas both
Yeah.
technology, they sometimes need that kind of technology, that expertise to tap into actual good market, and which, who knows, may become really big later at a later stage. So thank you,
Yeah.
Anupriya, for sharing your insight. Eve, do
No.
you want to add something?
Yeah. Could you repeat the question again? I understood Anupriya's answer, but did you
Mhm.
say how can Novonesis help
Mhm.
startups
Smaller
with?
companies, yeah, startup companies and SMEs, basically to help them with their technology, or how they can collaborate or in what fashion they can collaborate to bring more
Right,
market, bring good products to market, basically. So yeah.
right. I mean, there is the other competitor of Novonesis. I won't mention their name, but I approached them for some solutions recently and a while back. They do have solutions for startups specifically. So from
Okay.
what they said, so
Interesting.
I'm sure Novonesis, but it might be a completely different department, that deals with external
Yeah.
relations, or
Mhm.
yeah. But
Okay.
yeah, but that's for buying, as in if you want to buy the products of
Mhm.
for example, Novonesis, they'll give it at a significant discount, that okay, you'll be a future big company and I want to have you as my customer in the
Mhm.
early stage.
Mhm.
Um, but apart from that, I have seen a lot of like programs,
Mhm.
like startup challenges programs, like funding type programs, accelerator programs with large enzyme companies. I have seen that. Yeah. So maybe
Mhm.
that would be a way to collaborate.
Amazing. Thank you. Thanks for adding that, Eve. And actually, my next question goes to you and Shubhankar, as you both are from the startup universe. So there is a question from someone who says that what sort of advice would you have for someone from a non-scientific background who's planning to start a food product business with biomass fermentation component? I'm still learning the basic information for now, but curious as to what might be realistic to pursue at an early stage.
Right. I answered that saying GFI, talk to GFI.
I
That
see.
was my answer. But I wasn't kidding. I think there is a startup manual on GFI website. What do you do if you want to
There are
make a
resources.
new startup? Yeah. But beyond that, I would encourage to. I haven't looked at Novonesis, but now I will. I'm pretty sure there is something like, if you are a startup and you want to apply to some accelerator program, you can. So I would encourage to look at those. And since the question was non-scientific background, I would say find a co-founder who is solid in the science, because you will quickly go down a rabbit hole if you don't understand the science of it. The commercial comes,
Mhm.
you need to be patient. The commercial will come, but it's maybe years down the line. So the science matters. So a solid scientific co-founder is
Mhm.
needed. Yeah.
Great. Shubhankar?
Yeah, I think just inculcating the science into your team. So whether it is, if you're young enough, get a second master's degree or something, or if you're old enough and you're like, okay, I need to start something now, a science co-founder is good for a well-rounded team. Um, Also, I think maybe picking something that doesn't need a science co-founder, right? Like clearly understanding that you'll not go into a lab or get a collaborator to work with you. So, there have been some commercial sort of plays happening where there'll be sort of celebrities who have a branding marketing team who have their own money, will go to an expert or a big company and will get sort of a white label product made. And there's nothing wrong with that. It's just about finding the right. So maybe go talk to a B2B player, but I would say be very clear about where your strengths and weaknesses are, what to do and what not to do. So if you're like, I'm not going to step into a lab by myself ever, then maybe better to go with a company or go the longer route. This is of course more expensive depending on what your capital availability, what your background is like. But if you want to do the longer route, inculcating the tech is always going to be better in your team. It's just going to take you longer.
Great. Thank you, Shubhankar. Thanks for that insight. And with that, yes, I think we overshot our 25 minutes, but thank you so much for contributing your thoughts, your experience to all the questions. I'm so sorry if somebody's question has remained unanswered, but I'm sure LinkedIn is an amazing way for people to network. You can also reach out to JFI as well. And thank you, Eve, also for extending, like directly extending people to have a look at what kind of open source information that you can provide to them. Amazing. Uh, so yeah, with that, I just would like to again extend a heartfelt thanks to Anupriya, Eve, and Shubhankar for contributing to their time, contributing their time, insights for this webinar. All the audience for being so patient and asking amazing questions to our panelists. And yeah, the All Protein Dialogues webinar will continue. Yeah, sorry, Eve, would you like to say
Yeah,
something?
sorry. I do need to just inform everyone my LinkedIn is down. I'm trying to get it reestablished. It's not my fault. It's LinkedIn's fault. Please email me for any questions. I'm a real person. I exist. I spoke at the webinar, so I'm not on LinkedIn at the moment. So just wanted to say that.
Great. Thank you. Thanks for saying that, Eve. The LinkedIn URLs can be found on the registration page. So when you registered for the webinar, you would see speakers there along with their LinkedIn links already mentioned. But as we know, Eve's LinkedIn is down, so you have her email address to write her to, or wait for a few days until the LinkedIn's back and you can reach out to her. Once again, thank you everyone for being here. And stay tuned for our next All Protein Dialogues webinar, which will come in the next couple of months or so. But until then, have a great evening or morning or night, wherever you are. Thank you very much for joining in.
Thank you.
Thank you.
Bye-bye.
Bye.